冒頭にご説明いたしましたとおり、グラス博士の体調不良のため、今回、日本までお越しいただくことが叶いませんでした。でそこでグラス博士はですね、長年研究を共にした愛弟子でありまして、また家族ぐるみで、ファミリーでお付き合いのあるポール・スネルグローブ博士に、グラス博士の代理として日本に行くようにということで委託されました。テーマは A New Appreciation of Ocean Life ということで、海洋生態系についての新しい視点というテーマでご講演をいただきます。
Well, good evening, ladies and gentlemen, and thank you for coming. I spent today in Tokyo with my friend Hiro Tsutsumi and his wife, and they took me to the great heights of Tokyo at the Sky Tree. And so tonight I want to take you to the great ocean depths in contrast. And specifically I want to talk to you about ocean life. So I come from an island off the coast of Canada, Newfoundland, which experienced one of the worst fisheries collapses in human history. This photo was taken around 1900, which shows a little boy next to a codfish, which are largely gone today. When I was a boy, my grandfather would catch fish half this size, which I thought was normal. Today, fish are half that size again. Some 15 years ago, I learned for the first time that walrus were once abundant in coastal waters of Newfoundland. This is something I had not known, despite the fact I grew up in this place and had studied the ocean ecosystem there for over a decade. Our expectations of the oceans, our shifting baselines, change with every generation, so that we really don't know what we've lost, or more specifically, what we might be able to get back again. Most of us, including me, had thought that human impacts on the ocean go back some 50 to 100 years, not coincidentally our lifetimes. Now, many of you will recognize the International Panel on Climate Change, but you may not know about the International Panel on Biodiversity and Ecosystem Services. That panel addresses how biodiversity loss affects how life exists on Earth and how human beings will exist. The idea is that living organisms in the ocean, its biodiversity, support a whole range of critical functions within the ecosystem. And these are processes such as climate regulation and nutrient cycling that keep our planet working properly. These functions, in turn, support important ecosystem services, which are the outcomes that directly benefit us as humans. So these include things such as harvesting nuri, fisheries, and biotechnology products such as medicines. All of these functions and services connect to each other, and all these activities that we engage in sometimes threaten them. This new area of study asks whether we must sustain all marine species, since we don't know which species do what in terms of these critical functions and services. Keep in mind that researchers estimate that the oceans provide some 2/3 of the $33 trillion of the functions and services that we see on Earth. So, as a quick example, although most people don't make money from seagrasses, in my part of the world, cod fisheries are just as important as tuna fisheries to Japan. Now, seagrasses provide a space for baby codfish to hide from predators. So the seagrass species provide a key ecosystem function that supports a service that humans care a lot about, the fisheries. So if we lose the seagrass, we will lose at least some of the cod that help to feed us. And so Dr. Fred Grassley became fascinated by ocean life early in his career, which he devoted to understanding marine biodiversity, particularly the deep ocean. I want to tell you about a few of his key contributions, and then tell you about the Census of Marine Life, which is a major program he co-founded and inspired. So Dr. Gracia was an early advocate of using submersibles to place marine biologists on the seafloor where they could actually see with their own eyes what was going on. This approach also made it possible to do experiments on the seafloor to understand what factors were important for seafloor life. He therefore really started experimental work in the deep sea. So Dr. Grassley worked with Howard Sanders, the father of deep sea biology, who discovered that deep sea environments are among the most species-rich on the planet. But photographs of the deep sea reveal large areas of seafloor covered in sediments that look unlikely to support a rich diversity of life. So Dr. Grassley proposed the patch mosaic model, the idea that small patches of food create little habitats that support different sorts of species, And scientists today still work with that very same idea. Indeed, it was central to my own PhD work that I did with Dr. Grassley. In the early 1980s, Dr. Grassley was able to assemble the largest data set on deep sea diversity ever collected. And it revealed even greater biological diversity than even his mentor, Dr. Sanders, had thought. From this work, Dr. Grassley estimated that the deep sea contained 10 million species. Scientists today continue to debate that number, but importantly, people became very excited about the diversity of ocean life. Prior to the first discovery of hydrothermal vents, we thought that much of the deep sea looked like this. But when the submersible Alvin arrived at the hydrothermal vents near the Galapagos Islands, scientists were astounded by the abundance of life that they saw. No biologists participated in that original cruise. And so Dr. Grassley organized the first biological cruise to collect biological samples and understand these very special environments. Because they are such small areas of the seafloor, a submersible, Alvin, was absolutely necessary, and Dr. Grassley had already shown how submersibles could be used for marine biological research. In 1979, they brought back impressive specimens of meter-long tube worms that would change the way that we understand how life on Earth works. Dr. Grassley encouraged his colleague, Dr. Cavanaugh, who was then a graduate student at Harvard University, who would eventually figure out that hydrogen sulfide that spewed from the seafloor supported bacteria living inside these animals, and that therefore supported these vents. So these bacteria could convert organic carbon and thus feed the huge numbers of organisms that occurred in these hydrothermal vents. And so began the study of a whole new ecosystem that rewrote textbooks, fueled by chemistry rather than by sunlight, and quite possibly the origin of life on Earth. One technological development that is not widely recognized is Dr. Grassley's contribution to how we observe the oceans. He strongly felt that the way we studied the oceans in the past, mostly from short trips on research cruises, was incomplete because it missed important events in the ocean. In the 1990s, he therefore set out and was a very strong early advocate of the idea of sending fiber optic cables to the seafloor to provide power to oceanographic instruments and to transmit data back to shore. Cabled observatories are now widely used in oceans around the world, including here in Japan. Dr. Grassley was also a great advocate of data sharing, because no single scientist can sample enough of the ocean to answer many of the key questions on ocean life. He therefore founded the Ocean Biogeographic Information System, which brings together biodiversity data that researchers are willing to share in a single database. It now contains more than 30 million records of life in the oceans. And by records, I mean presence of a given species at a given geographic location. This combined data produces a global image of the known and unknown in the oceans. So here, the red colors in the image show the best known parts of the ocean, and the dark areas show you the poorly known regions. Not so surprisingly, the North Sea and Eastern North America are among the best sampled so far, and much of the ocean remains blue or black, indicating little or no biological data. This side view of the ocean that you see here on the insert shows you a cutaway version of the ocean, and you can see that in fact our knowledge is also strongly biased towards the surface layers and not very much of the deep layers beneath. OBIS is available to anyone in the world, including the people of Japan, to download information on ocean life free of charge, and represents a major output of a program called the Census of Marine Life. So Dr. Grassley took advantage of the renewed interest he had helped generate in marine biodiversity to co-found the Census of Marine Life, the largest international effort ever undertaken to study ocean life. It was a 10-year, $650 million study to understand and explain the diversity, distribution, and abundance of life in the oceans, past, present, and future. In a sense, it was a social experiment, supersizing science by bringing together experts from around the world to study a variety of problems about ocean life, making it a little different from most of the other international projects that typically focus on one question and one technology, because ocean life requires a whole variety of technologies. So Japan actually played a significant role in many of the Census of Marine Life projects, but the Nagisa Project in particular was conceived by Dr. Shirayama, the executive director of JAMSTEC here in Japan. It taught school children from around the world to sample the shoreline, and it created a global data set using school classes to collect standardized data from around these different parts of the world. The Census Program looked at oceans past to appreciate the rich productivity we've lost, such as these abalone that you see here in California that were once hugely abundant but are now an endangered species, and tuna that were once common in the North Sea of Europe and are now largely absent. We also looked at the ocean present to look for reasons to celebrate and also for reasons to be concerned. And then we also thought about ocean's future, what we want them to be and what we fear they could be if we don't act appropriately. Systematic collection of scientific data really only started after World War II or later. So what did the oceans look like before that? Census scientists looked at a variety of unlikely sources of data, such as ship logs, monastery records, and restaurant menus, to try to learn what people were extracting from the oceans and how much of that. To their surprise, they found that some declines began with the Romans, who discovered that preserving fish with salt, in the absence of refrigeration at that time, allowed fishermen to catch more than they could eat or sell that day. And so began industrial fishing, sometime around 700 BC, or possibly even earlier. This Tunisian mosaic that you see here on the right-hand side shows trawling in about 500 AD, so a very long time ago. So we now know that humans depleted some species thousands of years ago, not just our lifetimes, but thousands of years ago. Here, the dark green bars show historical depletions of 65 to 98% for different groups of organisms once humans arrived. But in light green, we also see recovery of some groups to near historical levels once we finally leave those organisms alone. So, for example, we see groups such as marine mammals and some seabirds start to come back. But for the most part, we've depleted many species in the ocean. Historical photos from an annual fishing contest in Florida show large species in the 1950s replaced by smaller and sometimes very different species by the 1980s. By 2007, the winning catches would have been laughed at by the winners of the 1950s contests. But these losses are really no laughing matter. And there are many other examples of how ocean productivity has been lost over the years. But our view of the ocean is becoming clearer because we now have better technologies, and this technologies holds great promise for better understanding and better management of the way we exploit life in the oceans. These tools give us opportunities to look into the bottom of the ocean, to give us eyes into a world far away that's far different from our land-based existence. Today, we can use sound waves to measure ocean currents and to count fish as they swim by. Because although light does penetrate seawater well, does not actually penetrate seawater very well, sound waves actually do. And we increasingly use non-optical tools to see and measure ocean life. New technologies bounce sound waves off fish and count their reflections so that it's now possible to image an area of ocean the size of Manhattan Island, shown here. We see about a quarter of a billion herring, and that was done in a period of about 1 minute. And so that technology has really advanced the way that we can quantify fish without actually hurting any of them. So we can get accurate counts quickly, automatically, without harming any fish in the process. Satellite tags attached to animals show us impressive migrations across the ocean. The GPS global positioning system doesn't work underwater. But for animals that surface to breathe, for example, this elephant seal, it's an opportunity to identify their position, shown here in blue circles, and send these positions back to scientists around the world so they can understand where those animals are moving. So we use satellites then to track their movements in the oceans. So we can track seabirds, turtles, whales, as well as fishes. Now for animals such as tuna that don't surface to breathe, we use something called pop-up tags, which pop up to the surface after a period of time, after they've accumulated data on both light and day length. They pop to the surface, transmit their data via satellite back to the shore, and scientists can then figure out where those tuna have moved. That information allows us to calculate daily locations for species such as tuna, so we can understand their migrations and why they migrate, and therefore manage their populations much more effectively. So census scientists about a decade ago decided they wanted to catalog information on all known species in the Gulf of Mexico in the United States. This labor of love had no clear economic payoff other than pure scientific knowledge and love of information. But then, of course, as you all perhaps remember, the Deep Horizon oil spill, that data suddenly became very relevant. And so their data will likely be a very critical element in trying to understand the impact of that oil spill and how quickly the Gulf of Mexico recovers from that spill. I use this example to illustrate the importance of baseline knowledge of ocean life in the context of unknowns. The often unpredictable changes that we see, the future changes, can therefore be better managed once they happen if we know more about what's out there. So knowledge sometimes helps us to plan for change. So the census found spectacular beauty just about everywhere we looked, from the shoreline to the deep ocean, from microbes to fishes, and everything in between. Marine scientists describe 4 to 5 new species every day, and that number is limited by taxonomic specialists who can recognize and name those species rather than the pool of unknown species, which is in fact still very large even after this 10-year Census of Marine Life program. Now, you might think that a lot of the species out there are small and insignificant, but in fact, new species discoveries come in all shapes and sizes. So, indeed, much of the new discovery is small organisms, but some of it, in fact, is not. While it's hard to imagine that scientists would not know of the existence of 3-kilogram lobsters off the coast of South Africa, in fact, fishermen less than a decade ago requested a permit to export these lobster, and scientists recognized they had a new species. Similarly, a 3-meter-long kelp found in Alaska is also probably a new species, as is this 7-meter-long bigfin squid captured from the deep waters above the mid-Atlantic ridge. Census scientists also discovered new species that live in Arctic ice that support an ice-based food web. At the other extreme, the Yeti crab, discovered near boiling hot hydrothermal vents near Easter Island, captivated the public alike and inspired artists and children. Recently, scientists have found the hottest vents at over 400 degrees Celsius, the deepest vents at over 5,000 meters, known so far at least, and new vents in the South Atlantic and the Arctic where none were previously known. New analysis of diversity hotspots shows that diversity of species living along the coastline peaks in the Indo-Pacific. This is driven largely by tropical corals and the species associated with those coral environments. But surprisingly, oceanic species appear to peak at mid-latitudes. Managers and conservation groups can use this type of information to try to identify priority areas for conservation. So this is one way that we can try to minimize loss, and also to do something when loss starts to happen. Census scientists also found spectacular distributional changes, such as a seabird called the sooty shearwater that completes spectacular migrations from New Zealand to Alaska and Japan, and then back again, as it pursues endless summer in an effort to complete their life cycles. So we now know a lot more about where animals move in the ocean and why they move in the ocean. Scientists also now use animals as oceanographic ships. It's a less expensive alternative to formal oceanographic ships. So what we do there is to attach sensors to marine mammals to collect oceanographic data as those animals swim around the oceans. So that data may be as simple as ocean temperature and depth, or it may be much more elaborate than that. This means we can get data from all sorts of distant areas where we simply can't afford to send research ships, but we can afford to collect the data that these different animals collect for us. Now, marine scientists recently produced new estimates of the known and unknown diversity of life on land and in the oceans, and they concluded that we know of about 14% of the species on land and probably only about 9% of the species in the ocean. That's the area that I show here in dark blue. These numbers suggest that some 2 million unknown species still exist in the sea. Another approach has lowered that number somewhat to something more closer to 1 million, but that's still a lot of unknown species. These are all just estimates, of course, but they are very likely correct within an order of magnitude, and so they do give us some idea as to what's left out there to do. And so we've lost a lot of ocean paradise. For example, the abundance of trash this deep sea trawl collected in the Mediterranean Sea actually exceeded the number of animals that were collected in that trawl. We think of the deep sea as the last frontier on the planet, which humans have not damaged, but in fact, we have. But there are still beautiful habitats in the ocean, and so there's still a lot worth preserving, such as this deep sea environment off the east coast of Canada where I live. So I'd like to finish up by saying that ocean life inspired Dr. Grassley's career, and his research taught us many things. He sends his heartfelt thanks for the opportunity with the JapanPrize, and that opportunity that it's created for me to tell you about ocean life. The importance of that life and what we can do to maintain that life. I think in the future, we must do better. And I think we can do better, and some of the information we've collected over the last decade, and thanks to Dr. Grassley's effort, put us in a position to do just that. Thank you for your attention.